Hey, it's Mr. YS, and this is a tutorial on the Meselson and Stahl's experiment to do with DNA replication and figuring out how does DNA replicate. So they had three different possible ways that DNA could replicate. They knew that the structure meant that down the middle you had weak hydrogen bonds. And so the separation would be into the two strands with the bases on the inside. And so this suggested a mechanism where if the two strands are separated, you can use each strand to make new strands. But that still leaves you with some different options. So we've got three different models here that could have been the way in which it was replicated. And in the first one, in conservative, you can see that in the next generation, if this is the parent DNA, well, down here in conservative, we have kept the original parent DNA altogether. So we've got this, which is 100% conserved. So this is called conservative replication because it's conserving the original DNA together. You've got 100% conserved, and then you've got 100% new DNA. So essentially you're taking the old DNA, you're using it as a template, and you're making a completely new double helix. So you get the existing double helix, and then the second completely new double helix. So the second one, which we now know is the case, is the semi-conservative. And in semi-conservative, if we follow the parent, well, we've got one parent and one new, one parent and one new. So each of these new DNA molecules is half parental and it's half new DNA. And that means that if we were thinking about the proportion of new and old, well, it's 50% new and 50% old or parental DNA, where each of the parent strands has been used as a template and it's combined with new DNA, and then these go off to make the DNA of the daughter cells, because this is going to happen in mitosis. Okay, the third one, which wasn't really that promising, was the idea that somehow, rather than keeping the strand intact, that maybe you copied it and you got bits mixed in. Some parental, some new, some parental, some new. And this was called dispersive. So that was the third one, but that was less promising. So one of the questions which came up, which was tricky to answer, was to compare and contrast conservative and semi-conservative replication. So see if you can do that. Stop and see if you can make a table where you have conservative and semi-conservative, and you compare them. So what could we compare them for? Or what are the major differences? Is there anything that they both do? And what are the differences between them? And so we can think about copying, and we could think about the results. So here's what the IB had to say about it, and it's kind of a repetitive answer. But in semi-conservative, in the end, the DNA is half parental. It's 50% parental and 50% new. Whereas in conservative, all the DNA is parental for the one copy, well, the existing copy, and then the other new one is all new, because you'd opened up the existing strand, copied, and made DNA that was all new. Now that is not correct, so we know that that is not what happens. What else could you say? You could say that one strand of the daughter DNA, one strand of each new double helix is new, whereas in conservative, and see it's just a repeat again, all is parental or all is new. And to allow that, that means that for the semi-conservative, both the strands of the parental DNA are separated to achieve this, whereas in conservative, both the strands of parental DNA, they remain together after the copying. So how do we actually know that that's the case then? What did Meselson and Stahl do? Well, they wanted to figure out where the strands were going. And to figure out where the strands are going, you need to track the DNA in some way. You need to be able to separate DNA. And you need to be able to separate the parental from the newly synthesized. Now, because you're only talking about one thing, DNA, we can't separate it by picking a different element in it. We can't do what the Hershey and Chase experiment did, where they took radioactive sulfur, which is in proteins, versus the radioactive phosphorus, which is in DNA, because we're only talking about DNA. So what they did was they took something that's in DNA, which is nitrogen, because we've got nitrogenous bases, and they grew cells in two different environments. One which had the normal nitrogen 14, N14, that's the normal one, and then one which had this heavy, they called it heavy N15 because it's more dense. And so N15, if it's more dense, if we have DNA that's grown in N15, it would become more dense than normal DNA. So the next principle here is that if we spin round tubes in a circle, if we centrifuge, then the things that are more dense will go to the bottom of the tube. And this is actually how they separate cell components. So if you burst open a cell and you want to separate parts, if you spin it round in a centrifuge, and they're doing this very fast, like 50,000 RPM. And when you do that, the heaviest bits, the densest bits, will go to the bottom. And so in a cell, this would be like the nucleus would go right down to the bottom. For the purposes of this experiment, though, we're thinking about separating DNA, which is slightly heavier, from DNA, which is slightly lighter. So we could talk about light and heavy DNA. So the DNA with the N15 would be heavier. It would separate into a band further down. And the lighter DNA would separate into a band further up. So we get this kind of banding. Okay, so that's important to understand. You can spin things around and you can separate them by density. And so if we can have the parental DNA being grown in a different medium, so we talked about mediums with Hershey and Chase. The medium is the kind of growth solution. If we have a different medium of growth for the parent DNA versus the new DNA, if we grow the new DNA in a new different medium, we can try and track what DNA has been put in. So that's what they did. They grew up the E. coli in two different mediums. So they grew up the E. coli in a heavy N15. So this was the denser medium. it contained nitrogen N15, the heavy isotope, and that meant that these are the generations here. In the first generation, which we call generation 0, because they grew it only in the heavy medium, if they centrifuged the DNA, so they had to break open the cells and centrifuge the DNA, they would get only one band. And this band was further down because the DNA was more dense. So this is our N15 band. So E. coli grown in N15, all the DNA that it makes, we can imagine all the nucleotides that it makes, and it's really going to be this part with the nitrogen, they're all going to be heavy. So we can label this as red. So when it makes its DNA ladder, the double strand made up of the two sides of nucleotides, every nucleotide will be heavier. And so that DNA will sink further down. If the DNA is made in the lighter N14 medium, then all the DNA would be lighter. So if we did that experiment and then centrifuged it out, we would only get a band up here. This would mean that it's 100% light. So that's the density, this band up here would be the density of 100% light, and this band down here would be if the DNA was 100% the denser N15. So they did the experiment, and the first density doesn't tell them anything. So this is the original growth medium. All of the DNA was N15. So this band doesn't tell you anything. It's the next band, because what they do after this first band is now they take the E. coli, the bacteria which has got its chromosome of DNA inside, and then they put it into a different growth medium. And actually, rather than being in a plate, it was probably in a test tube. They put it in a growth medium where the only available nitrogen was the N14. So this has only got N14 in it. And that means when this original E. coli, when the E. coli would copy its DNA, which was all heavy DNA, so we can imagine that as being all this heavy DNA, but now it's only got a medium where it can add in the light form of the nucleotide. So that's the only thing it can add in. And so what it would do is, in this growth medium, we would take the original parental double-stranded ladder, we would open it up, and then the only thing that could be added in is the orange, the lighter, and we would add that in on both sides. Okay, now once we've done that, if we now centrifuge that, where will this half orange, half red be? Well, if it's half orange, half red, it will be less dense. It's going to move up. So when they centrifuged the new generation, generation 1, they found that all of the DNA was slightly less dense. It had moved up. It was at this intermediate density. So that told them, that excluded one of the forms of replication. The fact that none of the DNA was at the original density. because for it to be at the original density, it needs to be all of the heavy N15. So if we apply it back to this first diagram of conservative, if replication was a conservative replication, then this would be the red. They copied it in the N15. And in conservative replication, what we should get then, if it was conservative, if we kept the parental strands together afterwards, is we'd get an N15 band where the parent had come back together, which means we'd get a band lower down. And then the other band that we would get would be the new lighter DNA. And this new lighter DNA should occupy a band that was further up. So if they'd got this, then yes, tick, it would be correct. It would be conservative replication. But they didn't get that. They didn't get two bands. So straight away they could rule out, we know it's not conservative replication, because what we've got is a band that's in between. It's this intermediate density caused by having 50% parental and 50% new. So if we colour that in, that would be the original parental was all the heavy N15, and then one strand would be heavy N15 for each, and one strand would be the lighter strand. And that's giving, because it's 50-50 between the heavy and the light, it gives us this intermediate band. Let's have a look how it plays out. So at generation 0, we've got 2 heavy. And then at generation 1, once we've split and copied, we've now got the 1 red and the 1 orange. So 1 heavy, 1 light. And that's for both of those double-stranded molecules. So that gives the band over here, which is intermediate. Now if we open those up and copy again, then what we're doing is we've got 1 orange existing, and then the only thing that's in the medium is the N14, the light form of nitrogen, so the lighter, less dense nucleotides. So we would add in another orange for this. But the red is still there, so the red is going to be, we're going to still have 1 red, and then we're going to add in another orange, because that's the only thing that can be added in. Now if we repeat on the other side, the orange existing N14 lighter strand is going to be added in with a new strand, which is still N14. And then we're going to add in 1 orange for the new strand and 1 red, which was the existing. Well, let's have a look at what we've got here, because we've got 2 different things. We've got these 2, which are the same as this, where they're this hybrid, the hybrid between the parent DNA and the new light DNA. So those ones are going to be in exactly the same band. They're going to occupy that same middle band for that intermediate density. But the other ones, these 2 here in the middle, they came from newly synthesized DNA in generation 1, and now we've added in more newly synthesized DNA. So all they've got is the 2 light. And so these are going to go up and occupy this. If we think about the ratio, we've got 2 out of 4, which are this new, completely new light N14. And then we've got 2, 2, which are the hybrid. And so in generation 2, they got this banding pattern, which was 50% was hybrid DNA and 50% was occupying the light band up there. And so what this did was it disproved the idea that it was a mix of DNA, that it was dispersive. If it was mixed in, then all of the DNA, so if these, rather than being, so this is the one that kind of disproves it, actually both of them do. If we've got red and orange, that means we've still got some of the original parent DNA. Whereas if it was dispersive and it was mixed in, where we had a little bit of red, a little bit of orange, a little bit of red, and that was the same for both strands, then it would all be the same density. It would all be the same density after each copying. Now, it was all the same density after the first copy, but after the second generation, we've now got these two bands, which shows it's not dispersive, that it's definitely semi-conservative. Here's our summary. So Meselson and Stahl, they did the experiments to find out the mechanism of DNA replication. They radioactively labelled, and really it was the fact that it was heavier. So N15, although it's radioactive, they weren't really tracking the radioactivity until they kind of examined the pellets. It was more to do with the density difference. So they used N15 versus the normal N14. So they grew the E. coli, allowing replication in only the heavier N15 environment. And then they centrifuged the DNA after one generation, after two, and after three. After one generation, all the DNA had an intermediate density, and that was because all the DNA was a mixture of the existing parental N15 and the new, so this new daughter strand, which was N14. So all the DNA had that intermediate density compared to DNA from either the N14 or N15 environments. But the next generation showed a band which had all N14 DNA as well as an intermediate band. So that's when we have this occupying one band, the hybrid intermediate, and then we got a new band where it was only, the new band, it's only the light DNA. And so therefore, the replication is definitely semi-conservative because one strand was parent and one strand was newly synthesized. And if it wasn't semi-conservative, we'd have got rid of this band. All of it would have just moved up. And if it was conservative, then after the first generation, so after the first generation, what I found was just an intermediate band. That intermediate band was in the middle. So they wouldn't have found that. And the final bit that we're going to look at is DNA sequencing. We'll come back to this. So in DNA sequencing, they need to figure out what bases are there. Now obviously DNA is too small to see. And so what they do is they do replication, they do essentially copying of DNA in an artificial environment. So if we wanted to know the sequence on a particular DNA strand, we would make it single-stranded. And we want to try and figure out what is the sequence on this single strand, because from that we can work out the sequence on the other strand. So they use a special mixture. They've got these special nucleotides. They're called D-ATP, GTP, CTP, TTP, although those names are not important. So these are di-deoxyribonucleotides. And they're special because what they do is they stop replication. They don't allow a new bond to be formed after. So as soon as one of these goes in, it stops the replication there. So they allow the sequence of DNA that they want to work out to mix in a medium where they've put in these special nucleotides, where as soon as a nucleotide is incorporated, which is one of these dideoxy, it stops replication there. A new base cannot be added afterwards because it's a block to replication. It's because you can't make a phosphodiester bond to a dideoxy ribonucleotide. Okay, so let's see if we can think about what's going to happen here. So the other point is that it's not only these that are floating around. You've also got your normal ATGC. So there are some normal ones floating around, and then there are these special stop replication nucleotides. So what's going to happen is through randomness, sometimes you're going to put the normal one in, and then you're going to put another normal one in, and then you're going to put another normal one in. And in that case, the first three are just going to be normal. But then through random chance, in the next one, maybe you put in one of these stop dideoxyribonucleotides. So in this copying, and there's a polymerase that they use in there called Taq polymerase, which is a special polymerase because it withstands high temperature. It can withstand 90 degrees, and they found it in hot spring archaea. So we looked at archaea, which are a form of prokaryote. So archaea are these hot spring bacteria, and they withstand high temperature. So they've extracted a polymerase which fits on and starts to add in nucleotides. So we got to this point. The first three, by random chance, were normal ones. And now up to the fourth one, well, the fourth one, by random chance, we put in this special stop. So replication has to stop there, because it can't extend the chain anymore. And that means that you get a fragment which is 4 long. There's 4 nucleotides there on that one. But on other ones that they've done, it might be by random chance that at the very first base you included one of these stop. So it stopped at only 1 nucleotide. And so through random chance, you will randomly incorporate the one that stops replication at all of these different lengths. You will get 1 base, 2 base, 3 base, 4 base, 5 base, until you get to randomly including the nucleotide which stops. So we're going to end up, by doing this, with a mixture, and it wouldn't just be this once, it would be thousands, millions of times. You would stop it there, you'd stop it at 1 nucleotide, 2 nucleotides, and so on. And this means that we're going to get a mix of different length chains all floating around in the mixture. They're not going to be nicely ordered like this. We're going to have longer ones, shorter ones, all different lengths. And so the next step is that they take all these different lengths and they put them into a gel, and they run them out in a process called electrophoresis, where the DNA gets attracted to a positive charge because DNA is negative. And so these start to move through, a bit like chromatography. So they're moving through, and then they will separate by size. So if we think, what's the smallest fragment that we've got up here? What's the smallest? The smallest will move the furthest. Well, the smallest fragment that we got would be just this one. We would have only the A and the T fragment. So this fragment would move all the way down, and there we go, it's the T. So our one base pair fragment would be the thing that moved the furthest, and then the thing that couldn't move because it's the largest would be our however many bases that is, but it's a G at the end. Okay, so we've separated them by size. And this is kind of weird that you can separate all the way down to 1, 2, 3, 4, 5, 6. And they actually run out, they've got something which is like a marker. They have things which they make in the lab which are 1, 2, 3, 4, 5, 6, all the way at bases long. So they've got kind of like a ladder that they can read it off from. And the next thing is, how do they see it? So now you've got DNA fragments which are 1, 2, 3, 4, all the different number of bases long. How do they see it? So this is where the special nucleotides come in. So these were the dideoxyribonucleotides. So the special thing was that they stopped replication, so we can get those fragments. But the other thing is that they fluoresce. When you shine a light at them, so if there's a light coming down at them, they will fluoresce with a particular colour. And they can do for each of these different ones, the A, T, G, C, you can see T they're showing, it has a red fluorescence, whereas G has a yellow, and A is green. So they, in a special machine, a sequencer, they shine the light at it, and now they know, okay, they've separated it by size, they know this is one base pair long. So for one base pair, the nucleotide, if they shine and it comes back red, then the computer can read that, it knows it's a T. And they shine the light again, and the next one was red, so it's a T again. And now they can figure out what was the sequence which paired to the template strand. And so to figure out the actual sequence on the DNA, we have to reverse it, because whatever sequence they were reading, it was made through complementary base pairing with the original DNA strand. So you take that and then you just work back. So if it was a T here, now we know in the original it was an A. If it was a G at the end, in the original it must have been a C. Here's where we're going to do the application bit. So stop and just read through the question and see if you can work out what the answer is. Okay, so let's have a look. They're saying that these sequences were terminated with the dideoxyribonucleotides, and they've run out these. And so because they've given us this bit here, it means it starts here. And you can see it's dideoxyribonucleotide. We've got A, T, C, and G. So these are basically the bases. This is an A, a T, a C, and a G. So which is going to be the smallest? The smallest will be down here at this end. And that means that the first one base pair will be the smallest. The first letter, which was read off, was an A. And if the first letter read off was an A, then it means the opposite letter of that was the first letter of the actual DNA template. So by reading that, we can straight away exclude all of those, and we know that the answer is this one. And let's just see if it follows along. So it's saying, because we know the first letter is T, then the second letter was C, so the opposite of this second letter, because then it's a C, but the opposite for the template is G, yeah. TG. Okay, done.